Human cloning, the process of creating a genetically identical copy, the human, has long been a topic of intense ethical and scientific debate. The prospect of manipulating life at such a fundamental level raises profound questions about the nature of humanity, individuality, and the boundaries of scientific advancement. Greetings, and welcome to the latest episode. If you haven't subscribed to the channel yet, be sure to hit that subscribe button now. In response to these concerns, various countries and international bodies have grappled with the idea of imposing a ban on human cloning. Human cloning involves the replication of an individual's genetic material to produce a duplicate. There are two main types of human cloning: reproductive cloning and therapeutic cloning. Reproductive cloning aims to create an entire organism, essentially bringing a cloned human into existence. On the other hand, therapeutic cloning focuses on creating cloned cells for medical purposes, such as tissue repair or organ transplantation. Critics argue that cloning undermines the unique essence of human beings and violates the inherent dignity each person possesses. The idea of creating individuals for specific purposes, whether for reproduction or organ harvesting, raises ethical questions about the commodification of life, the long-term effects of cloning on the cloned individuals, and the potential for unintended consequences pose significant ethical concerns. The scientific community remains uncertain about the health and well-being of cloned organisms, raising questions about the responsibility of scientists to ensure the safety of their creations. Cloning, especially if used extensively, could lead to a reduction in genetic diversity. This reduction might make the population more susceptible to diseases and environmental challenges. Maintaining genetic diversity is crucial for the adaptability and survival of a species. Cloned individuals may face social and psychological challenges, including questions about their identity, uniqueness, and acceptance by society. The potential stigma attached to being a clone could have profound effects on mental health and well-being. The United Nations has engaged in discussions regarding human cloning and its ethical implications. In 2005, the UN adopted a non-binding declaration urging member states to adopt measures to prohibit all forms of human cloning incompatible with human dignity and the protection of human life. Many countries have taken steps to regulate or ban human cloning. For example, the United States has no federal law specifically banning cloning but prohibits the use of federal funds for cloning research. In contrast, countries like Germany and Canada have explicit laws prohibiting reproductive cloning but allowing certain forms of therapeutic cloning. The approaches to human cloning vary globally. Some countries adopt a complete ban, while others allow limited forms of cloning for research purposes. The diversity in regulatory frameworks reflects the ongoing struggle to balance scientific progress with ethical considerations. Cloned animals often exhibit variations in gene expression patterns due to a phenomenon called epigenetic reprogramming. Epigenetic changes can impact the cloned individual's traits and behavior, highlighting the complexity of cloning outcomes. Cloned animals often show signs of premature aging, which may be linked to altered telomere length. Telomeres, protective caps at the end of chromosomes, tend to shorten with each cell division. Clones, derived from cells with shortened telomeres, may experience accelerated aging. Some scientists have explored the idea of cloning extinct species, including ancient humans like Neanderthals. While the ethical implications are profound, the technical challenges and uncertainties regarding the recreation of ancient genetic material present significant hurdles. Cloning can potentially lead to a reduction in genetic diversity, raising concerns about the resilience of populations in the face of environmental challenges. Maintaining genetic diversity is critical for species survival and adaptation. Cloning relies on the transfer of nuclear DNA, but mitochondria, the cell's energy producers, have their own DNA. Cloned individuals may inherit mitochondria from the egg donor, leading to a mix of mitochondrial DNA. This mitochondrial DNA mismatch can contribute to health issues in cloned organisms. Different countries have varying stances on human cloning, contributing to a patchwork of regulatory landscapes. While some countries explicitly ban all forms of cloning, others permit certain aspects for research purposes. This diversity in regulations reflects the ongoing struggle to establish a global consensus. Cloned animals may have a higher susceptibility to genetic abnormalities and disorders. The cloning process can introduce errors, leading to health issues in the cloned individual. This aspect raises concerns about the well-being of cloned organisms. Beyond the scientific challenges, the ethical implications of human cloning are profound. Questions related to individual identity, the purpose of cloning, and the potential societal impacts remain central to the ongoing discourse. Telomerase, an enzyme that maintains telomere length, plays a crucial role in the aging process. In cloning, the activity of telomerase is often disrupted, contributing to premature aging in clone organisms. Cloning remains a subject of fascination and concern for the public. The idea of creating genetically identical individuals has sparked debates about the implications for societal norms, family structures, and the perception of cloned individuals. The debate over human cloning is likely to persist as technological advancements continue. Ethical considerations, coupled with the potential for scientific breakthroughs, create a complex landscape for policymakers, scientists, and the public. Striking a balance between promoting scientific innovation and protecting ethical principles will be crucial in determining the future trajectory of human cloning. The call for a ban on human cloning arises from profound ethical concerns related to human dignity, unpredictable consequences, genetic diversity, and social implications. International efforts, both in the form of declarations and national legislation, aim to navigate the delicate balance between scientific progress and ethical considerations. As technology advances, the ongoing dialogue surrounding human cloning will shape the ethical boundaries of scientific exploration and innovation. Remember to hit the subscribe button, hit the bell button for new videos, and give us a thumbs up if you enjoy this information. And stay tuned for more enlightening content in the future.